Okay, Julia, let's talk about your new protocol. First, I want to review what was the original protocol and exactly why we're switching to the new protocol. The bacteria that we are incubating onto the surfaces overnight at 37 degrees Celsius and then swabbing the surfaces, that's going to remain pretty much the same. The swabs were going to be placed in LB broth and then broth incubated for 24 hours to see how much bacteria would grow on that amount. And then the absorbance was going to be measured to relate to turbidity compared to a normal LB broth blank. The problem is that what absorbance is measuring at that point is the comparison of how much is growing in the broth, not the amount that was growing on the surface itself. So yes, the original colonization amount that you swab off the surface would affect how much growth occurs, but it wouldn't affect it that much. And the amount that you would get from 24 hours of growth in a broth is staggering, and the differences between the two would not be very great. So we're going to move into a new protocol that deals with these. This solution is just good old basic counting colonies. Each visible colony on the agar plate is started from one living bacterium, meaning that one bacteria planted itself there and kind of just started to populate. And as long as we can count distinct colony-forming units, these distinct circles here, then we can assume that that was the amount one bacteria came from. Counting colony-forming bacteria units lets us measure living bacteria in real time, represented from the surface that we actually swabbed. Furthermore, we can compare surfaces directly because we can have different plates for different surfaces. It's a lot of counting, but I think I've made it in a way that will be easy for you. So let's talk whole process. So the revised approach that we're doing is going to apply bacteria to surfaces, incubate at 37 degrees Celsius for 24 hours, the same as before. This 24 hours is negotiable, as in you can change that if you like. That's just what I recommend because 24 hours growth is really easy. You start it on a Monday, you pick it up on a Tuesday. Then we're going to swab with sterile swabs equal sized surfaces after growth, which is the same as before. The size surfaces that you have set up are great because they're already equal sizes because you already measured all the surface area that you wanted. This is where things get a little bit different. We're going to transfer the surface bacteria not into LB broth, but a different liquid. And we'll talk about what liquid I think would be best in a moment here. We're then going to serial dilute the liquid to give us various concentrations. We'll talk about why that is later on as well. But what you're going to do is take a little bit of the stuff that has the bacteria and put it into another tube with a little bit less bacteria, and a little bit less bacteria, and a little bit less bacteria. It's going to be easy. It's super duper easy. I promise you, I've broken it down way, way too easy for you, okay? Then, each of those different concentrations of bacteria, you're going to place a tiny amount of each of those into the agar plate to let it grow. We'll spread it onto the plate, spreading it like we did with the yeast way back when, and we'll grow those colonies overnight. Again, negotiable. I still recommend 24 hours. If it's the case where you start the original surface on the Monday and swab it up on a Tuesday and plate it on Tuesday, then you can actually just have me take them out of the incubator 24 hours later at the Wednesday mark, and I'll put it in the fridge so it stops the growth for you. So you keep the consistency and can then count on a Thursday instead, which is the last step, counting colonies. Okay? Now, there is a little bit of prep work you have to do in order to get your plates ready. You have to make the plates. So you will make agar with water, agar powder, and LB powder, then sterilize it in the autoclave. Then, after it's sterilized, you pour that liquid gold, that gooey molten agar, into new petri dishes. Either the big petri dishes that we have normally, or the small ones that I got so you could have smaller ones available. Let me get that for a second. Got them right here. Now, you can see one is much bigger than the other. I have a small handout that will walk you through the whole thing so you know exactly what to do. Riley and Leah have done a bunch of these already. Morgan and Aiden have done a bunch of these already, so do not worry. It's super easy. It takes about 1 class period to make the agar, and then about 1 more class period to pour the agar. So you might want to, on a day where you don't have much to do, on a Thursday, Friday, maybe make the agar on a Thursday and pour them on a Friday or something like that. But because I want you to get started as soon as possible, I will make the first round for you. I will make 20 of these large plates and 30 of these small plates so you can decide which ones you want. You're welcome. The sterile saline here is super duper easy to make as well. You mix 0.85 grams of NaCl into 100 milliliters of water, and boom, you've got 100 milliliters of sterile saline. You sterilize it in the autoclave, of course, just like you did with LB broth. Now, why in the world are we making sterile saline? Well, that's the liquid we're going to put the swabs into so that the bacteria don't grow like crazy inside the tube in the time that we are collecting them, but so they don't get all terrified of the environment that they're in. Because bacteria like a little bit of salt. They're used to being inside your body. Your body has a little bit of salt, so therefore we'll put them in a salty environment. That way they don't burst whenever they hit a bunch of water. Okay, now quickly, step 1, growing bacteria on the surface is super easy. You're going to have each material exposed to B subtilis at the same time. You already made that liquor culture. You're very good. I just wanted to remind you of a couple of things to keep in mind as you go forward. That all materials should get the same amount of bacteria, which means you should determine how much liquid culture is spread onto the surface, or if you're using a swab to swab it onto it, how many times will you swab it, and you should incubate it for the same amount of time no matter what, because you don't want the plastic one to go for 48 hours and the stainless steel to only go for 24. You won't be able to compare the results. This allows the bacteria time to attach to the surface and grow if possible. We don't know whether or not these surfaces will allow bacteria to grow or not, so keep that in mind going forward. Step 2, you swab the surfaces. All surfaces should get an equal area of surface that is swabbed. And some consistencies to keep in mind. You want the same pattern of swabbing, whether it's like 10 swipes horizontally and 10 swipes vertically, or if it's like 30 seconds of swiping like crazy, that's fine. But again, you need to keep the same amount of time, the same pattern, the same pressure. Don't press too hard. Don't not press at all. Keep that same pressure on the swab onto the surface as much as possible. Try to keep these things consistent because you are applying this swab to the tubes in a second. Then you're going to take that swab, release the bacteria into the liquid. Literally, you're going to dip the swab into the sterile saline and allow it to be suspended inside of it. So you can dunk it in and press it against the sides to get all the bacteria goodness into the sterile saline. You can rotate it inside. You can spin it around. You can swirl the swab inside the liquid. You can even break off the sterile saline into the tube, break off the swab into the tube and vortex it for a fixed amount of time, and that would keep it consistent across the board. It's all up to you. You just have to figure out how you're going to release the bacteria from the swab into the liquid. You should normalize how long the swab sits in the liquid. That's something you gotta decide. Are you going to swab for 10 seconds and then let it sit in 10 seconds, or 20 seconds, or 30 seconds? I know you're petrified of making wrong decisions. There's really not wrong decisions here. So please try to be as decisive as possible. One thing you also want to keep it consistent is the same amount of liquid that you're dipping these swabs into. If you decide that you want 1 milliliter of solution, that you're starting off with, then that's 1 milliliter in a 15 ml tube. But there on out, you should use 1 milliliter for every single tube that you collect and dilute in. So I would suggest 1 milliliter just to make the math easy on you. You discard the swab after you're done with the transfer, and you should absolutely label the tube with your name, the date, and some taglines so that you can tell me what kind of bacteria it is, what material was swabbed, whether or not you had wear and tear on it, because you'll end up doing that as well. And you should label it as tube A, and this will be labeled with 10 to the power of 0, meaning that this is the original concentration. This is the dilution factor that we'll keep track of for our calculations later on. Don't worry too much about the numbers. I'm going to tell you exactly what numbers to write down. Don't worry. Step 4, the serial dilution. The thing everyone is scared of, but you're going to see how easy it is. You're going to set up 3 more tubes, just like the one before. But this time, instead of 1 milliliter of sterile saline, you're going to put 900 microliters. That is 0.9 milliliters of sterile saline. And you're going to label them as tube B, tube C, and tube D. Then you're going to take 100 microliters, 1/10 of the amount from tube A, and put into tube B. Then you cap it and mix it all up in your hands. Label that tube the same as tube A, but this time it's going to say tube B, and the serial dilution factor will be 10 to the first, because you just took 1/10 of tube A and put into tube B. Then you're going to transfer 100 microliters from tube B and put it into tube C. Cap it and mix it just fine. And tube C will be labeled with 10 to the second, because this is 1/100th. 10 to the second is 100. 10 to the second from tube A, the original one. It's 1/100th of tube A's concentration. Finally, you're going to transfer 100 microliters of tube C into tube D. Cap it and mix it. Tube D will be labeled as tube D, and it'll have this serial dilution factor of 10 to the third. That is 1,000. This one is 1/1000th of the same concentration as tube A, which means we should have 1/1000th number of bacteria, right? That's going to be a small amount. There's going to be a reason for that later, I promise. You will now have 4 tubes for that one material, all with the same B subtilis, just different concentrations of it. We then go ahead and plate those dilutions onto separate plates. Now you have 2 options for this. One is the one I recommend. You can pipette 100 microliters of tube A onto the plate labeled plate A, because it's going to be the same concentration as tube A. You'll want to label it with more than just that. You'll want to label it with the date and your initials, B subtilis, because that's the organism you're using. Maybe you want to put the serial dilution factor in. You definitely want to label it with the material. Keep your labels consistent with everything you do. The only difference between this is we took tube A and put it onto plate A. So label plate A exactly the same. Once you've pipetted 100 microliters of tube A onto the plates, you want to spread it evenly across the platform. Literally, you can just take it in your hands and rock it back and forth like this. Or I could sterilize some yeast spreaders for you. I don't think that'll be necessary. We could use sterile-tipped pipettes or sterile-tipped swabs or something, but don't get too fancy here. This rolling of your hands like this is good enough to get that smeared all over the top side. Now this 100 microliters of liquid that you're pipetting on would need to be smaller if you decide to go with these smaller plates that I'm making you. So keep that in mind going forward, that you might have to change this amount right here. That amount is totally okay to change, but it's up to you. We'll incubate it overnight. The other option for plating your dilutions is the more classic way that you know, and it's simpler. You just dip a loop into tube A and streak it in a tight zigzag across one area of the plate. It's not going to be a quadrant streaking because we're not looking to isolate these colonies. It's just a zigzag across in tight form, and then you incubate it overnight. Personally, I think you'll have an easier time counting with this option up here, pipetting instead of dipping. I think when you swipe, those colonies tend to stick together a little bit more, and it makes it hard to count. You're going to repeat this with each and every tube, so you should have how many plates? 4 plates, plate A, B, C, and D for each material that you do. Label everything. If you don't label stuff, you will get confused, and you will have no data to record, and you'll have to do it over again. Label more than you have to, that you think you have to. It'll make your life easier. Now, I wanted to show you this picture to help you understand exactly why we are doing this dilution. Take a look at these colonies up here for the S mutanans. Tell me, could you sit there and spend a whole hour counting each of those individual colonies? Sure, you could. But why would you have to? Why would you spend all your time counting all those colonies? We do a dilution so that we can count the colonies at a lesser concentration a little bit easier. It would even be easier to count it. This one was the original. This one was the 1/10th amount. This is 1/100th amount of concentration. And yeah, I could sit here and say, hey, 1, 2, 3, 4, 5, 6 colonies. I probably want to aim for one that looks like it has 30 to 300 colonies. And that will work out just fine for us. And that will be able to be put into some math so that we can find out exactly how many colony forming units we would have expected if this was the original colony concentration. So, step 6 is just to count the colony forming units. After 1 day of growth in the plates, you're going to pick the plate that has 30 to 300 colonies. And you only have to count 1 plate per trial. If you used my method of pipetting 100 microliters onto the big plates, then you can use this formula to estimate your bacterial concentration. The CFUs per milliliter will be equal to the colonies that you counted, how many colonies you had, divided by 0.1, that's 0.1 milliliters, just like that, times 1 divided by the dilution. The dilution is the number 10 to the x that we found in the tube itself. So if you decided to do the one that we diluted the 3rd time, that would be 1/100th, that's 10 to the 2. And we put 10 to the 2 in this denominator here. So it'll be a 30 to 300 in this number, and a 10 to some number, to some power, I should say, in this number. And you just do some multiplication, division, and boom, you've got how many colonies could form in how many milliliters of solution from your swab surface, which is awesome. This is the easiest way to possibly measure the number of bacteria that you have. I do want to emphasize that you could, and this is optional but recommended, create some controls for yourself and measure those as well for bacterial colonies, just to make sure that your project is going as planned. We could do a negative control by swabbing a sterile surface, diluting that and plating that. We should expect 0 colonies in that. That's what a negative control is for. Are we indeed sterile in what we are doing? We also can do what is called a process control. A process control double checks that our process of making sterile saline and all these steps of taking the swab and pressing on the sides or vortexing it is going to result in no colonies, because we want the sterile saline to be no bacteria. So we would just plate 100 microliters of sterile saline alone and expect 0 colonies from that. It's another negative control. The positive control for this, we could plate 100 microliters of your B subtilis LB broth that you have right now, and we should expect a ton of colonies to form. It's going to confirm that our bacteria can grow under normal conditions instead of being placed on a surface and then swabbed up and then diluted and yada, yada, yada. This confirms that our bacteria is normal. One last thing to remember is that you have to repeat this for multiple trials. So let's say next week you swab your bacteria on top of the surface on Monday, and then on Tuesday you swab it off that surface and collect that data and put it onto plates. And then on Wednesday I yank them out of the refrigerator or out of the incubator and put them in the refrigerator so they stop growing. And then Thursday you go ahead and count your colonies. That's all great. But you got to do that 3 times recommended each surface. So you're going to be counting maybe 4 plates each time because you want to do the 4 materials at the same time maybe, right? This is a lot of serial dilution. You got to get good at this. You got to practice because this is going to be a lot of time if you're slow and you aren't reading what you're doing. So read this over and over and over again. Furthermore, that does mean that you have a decision to make. Do you have 3 trials per surface? Do you have 4 materials? Do you have a normal wear and tear of nothing sanded down and then 1 surface sanded down at, 80 grit I think it was, and then another for 2,000 grit? And then finally, you got to think about how many plates you got to make, because you got to make the agar after this. I'm just making you 20 big ones and 30 small ones. You got to decide, do you want the big ones or do you want the small ones? And in that case, you'll have to make 4 plates for each trial. You have 3 trials. You have 4 materials. You have 3 different wear and tear trials. That's 144 plates altogether. And finally, do you want to have those controls I just mentioned? That's 147 plates altogether. So this plate making will go by a lot faster if you choose the small one, but it might be hard to count the colonies on a smaller plate because they'll be closer together. You might get less accurate results on smaller plates than you would bigger plates. So it's kind of up to you. Do you want to make a ton of big plates or a ton of small plates? And do you want better suited math or less suited math? Entirely up to you. I support you either way. I'll be there to help you out every step of the way. I'm already starting with your LB agar right now with the plates and stuff. I've got some made and I got your sterile water going, or the sterile saline solution going, so I'm helping you along the way. You just got to communicate and you got to decide. Okay? Let me know I could help. Hopefully this video made a whole bunch of sense to you. Please email me with questions over the weekend. Please think about the decisions you want to make. Please tell me about it. This is part of your check-in. Are you making the decisions on what you need to do for your project? Okay? If you don't email me this weekend and you don't think about it this weekend, I'm not going to give you a very good grade in your check-in. So, think about it. Check in with me. Cool.